Hello everyone, my name is AJ Keefe, and in this presentation I'll be covering the paper Nucleus-Translocated ACSS2 promotes gene transcription for lysosomal biogenesis and autophagy, published in the journal Molecular Cell. So I want to extend a special thanks to the authors for their contribution to this field of research. And for this video, I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before actually playing my commentary. And if you have any questions, please comment them below and I'll respond. And I hope you enjoy the video. Before getting into this paper, there are a couple of housekeeping items we need to review. So this paper centers most critically on these three main proteins, these three main pathways, TFEB, AMPK, and ACSS2. So let's start with TFEB, which is going to be this graphic. So TFEB is a master autophagic and lysosomal biogenesis transcription factor that specifically recognizes clear elements in DNA, CLEAR elements. And these clear elements are found at autophagic and lysosomal gene promoters. So the canonical mechanism of activation of TFEB is illustrated in this graphic. So where mTORC, we have mTORC right here, when it's activated at the lysosome, it constitutively phosphorylates and inhibits TFEB. So this phosphorylation seen right here simply produces a binding site for 1433. 1433 is an inhibitory phosphoserine binding protein that binds to certain phosphorylations, such as this one on TFEB, placed by mTORC, that prevents it from translocating to the nucleus. So recall that mTORC is an incredibly important kinase that is activated by activated by growth signaling and deactivated by starvation. So when mTORC is deactivated by starvation, or by an inhibitory phosphorylation, such as from AMPK down here. We're going to be talking about that in a second. So mTORC can be deactivated by starvation and by phosphorylation by AMPK. When it's deactivated, it's no longer able to phosphorylate TFEB, and the loss of inhibitory phosphorylation on TFEB allows it to enter the nucleus and prime lysosomal and autophagic gene expression. Another route of activation of TFEB, other than simply mTORC inactivation, is transient rises in calcium flux. So this mCoL1, this protein right here, can release calcium from the lysosome, and this can activate this CN, or calcineurin, which is a calcium-activated phosphatase right here, that can remove the phosphate from TFEB and it unmasks the nuclear localization signal of TFEB, which enters the nucleus and activates gene expression. So there's a few different ways of activating TFEB, but if TFEB is in the nucleus, then we know we have autophagy gene expression. So another key player in this process, as already mentioned, is AMPK. AMPK is a kinase that senses the ratio of ATP to AMP. So it's able to act as an AMP sensor because it actually contains an allosteric binding site for AMP. And in fact, this kinase, AMPK, requires AMP in this little binding pocket for its kinase activity to actually be functional. So AMPK not only requires AMP, but it also needs to be activated by a phosphorylation by either of these two kinases. We have liver kinase B, LKB1, and this calcium-activated CAMKK beta. So these two kinases aren't discussed in this paper, so I won't go into it, but either way, once AMPK is activated, it begins mounting a cell's response to starvation. And it does this most critically by inhibiting mTORC. So our AMPK would be right here. It comes in, it phosphorylates mTORC and inhibits it, prevents this inhibitory phosphorylation. AMPK also activates ULK1, ULK1. And ULK1 is the autophagy pre-initiation complex that, when phosphorylated by AMPK, can begin activating autophagy. So the last critical protein to understanding this paper is ACSS2, which is simply an enzyme that converts acetate into acetyl-CoA. So the conversion of acetate to acetyl-CoA is a incredibly important biochemical process because much of a cell's acetyl groups are locked in acetate molecules. And in order for these acetyl groups that are locked in acetate to be used for energy or protein acetylation, the acetyl groups need to be linked onto coenzyme A that acts as a hook and holds acetyl groups. And that's what ACSS does, is it hooks acetyl groups from acetate onto coenzyme A. So once acetyl-CoA is activated by ACSS2, it can then be used for energy by shuttling it through the citric acid cycle. Or alternatively, the acetyl group can be used for protein acetylation, including the acetylation of histones. So that's something to keep in mind, is that these acetyl groups that are activated by ACSS2 can be used for the acetylation of histones. And remember, histone acetylation is almost always an activating modification because it loosens the electrostatic interactions between DNA and histones by masking the positively charged lysines on histones that are attracted to the negatively charged phosphate backbone of DNA. And so the current paper discovers that ACSS2 is much more dynamic and interesting than a simple acetate to acetyl-CoA converting enzyme, and actually plays a critical role in autophagy. Because it turns out that when ACSS2 is phosphorylated by our AMPK kinase during starvation, it translocates to the nucleus, and in the nucleus, it's able to promote autophagic promoter acetylation. And also just note in general that when I say autophagic genes, I'm actually talking about both autophagic genes and lysosomal genes, because they're both driven by many of the same elements in their promoters, such as and transcription factors, such as TFEB. So that's just something to keep in mind. Anyways, let's get into the paper. So this ACSS2 protein that simply converts acetate into acetyl-CoA is known to be characteristically upregulated in hypoxic tumors. And often the depletion of ACSS2 inhibits the tumor's growth, suggesting that ACSS2 is in some way critical for hypoxic tumor growth. And another pathway that is uniquely important to hypoxic tumors is autophagy and glucose starvation. Glucose starvation actually triggers autophagy by activating AMPK, and it also inactivates mTORC and drives the translocation of TFEB to enhance autophagic gene transcription. So the question is there any connection between these two processes? Or more specifically, does ACSS2 promote hypoxic tumor growth simply by converting acetate into acetyl-CoA? Or is there a deeper relationship between ACSS2 and the general starvation response that is, autophagy? And it turns out, according to this paper, that there's actually a very profound relationship between ACSS2 and autophagy. So first, in this graphic, you'll see that ACSS2, right here, ACSS2 is targeted for phosphorylation by AMPK following energy stress. So AMPK comes through, it phosphorylates ACSS2, and phosphorylated ACSS2 unmasks a nuclear localization sequence, an NLS, that recruits and binds Importin alpha 5, or I'm just going to call it Importin A5. So Importin A5 then imports ACSS2 into the nucleus, where it binds to TFEB and is localized to autophagic genes, and it upregulates acetyl-CoA, and it upregulates histone acetylation at autophagic genes. And so this TFEB, which is known to localize to autophagic genes and upregulate their expression, also associates with this acetyl-CoA producing ACSS2. And so it creates kind of a local pool of acetyl-CoA for the acetylation of nucleosomes and histones. So it actually ends up upregulating autophagic and lysosomal gene expression. In fact, ACSS2's acetyl-CoA generation in the nucleus, according to this paper, is required for glucose deprivation-induced autophagy in these glioblastoma cells that they're using. They're using glioblastoma cells. This is amazing, right? That the ACSS2's acetyl-CoA generation, so its translocation to the nucleus, is required for glucose deprivation-induced autophagy in these glioblastoma cells. And I thought that was quite amazing, because preventing ACSS2's nuclear import or acetyl-CoA generation, or even phosphorylation by AMPK, preventing any of these activities prevented autophagy upregulation following starvation. So the movement of ACSS2 into the nucleus is an incredibly important process. And also just note that energy stress, we have this other arm of the pathway. The energy stress inhibits mTORC, which normally is phosphorylating TFEB, so it can bind the inhibitory 1433. When mTORC is no longer placing these inhibitory phosphorylations on TFEB, it's released, allowing it to be imported into the nucleus, where it primes gene expression. ACSS2 translocates to the nucleus during glucose starvation. So this was essentially the first thing that the researchers observed, was a massive translocation of ACSS2 from the cytoplasm to the nucleus during deprivation of glucose. So this is illustrated by this Western blot down here at the bottom. You can see that when glucose, or GLC, is removed, only about 5% of ACSS2 remains in the cytoplasm. So when glucose is negative, it's removed, only about 5%, 0.05, remains in the cytoplasm, and only 95%, 0.95, is now found in the nucleus. You can see when glucose is present, there's no ACSS2 in the nucleus. And we know we're looking at the nuclear fraction because there's no tubulin down here. No tubulin down here. So, and we also know expression doesn't change because the whole cell lysate, WCL, there's no change in total expression of ACSS2 in the whole cell lysate. So this can't be explained by change in expression either. So I also cropped out the bottom of this Western blot. There's a few more down here. But there's the other part of this Western blot simply showed that there was also no pCNA or in the cytoplasmic fraction. So pCNA is a polymerase sliding clamp that's only found in the nucleus, and that's another way they knew that they're looking at the nuclear fraction, is because the pCNA was present down here. Anyways, they next wanted to elucidate the mechanism that was triggering ACSS2 translocation to the nucleus. And so they started with AMPK. They started with looking at AMPK because it's simply known to be a critical starvation activated kinase. So they found that small molecule inhibition of AMPK or complete knockout of AMPK completely blocked ACSS2's translocation into the nucleus during glucose deprivation. So after observing that ACSS2 was being phosphorylated by AMPK, the next step was to try and find the site of phosphorylation, and they landed on this serine 659. So we're going to be seeing this S659A quite often. And so this serine 659 of ACSS2 is highly evolutionarily conserved. And they found that mutating this residue, completely blocked the nuclear translocation of ACSS2, as illustrated by this figure up here. This figure is based on the immunochemistry of images such as this. So you can see that when ACSS2 is wild type, we see no nuclear ACSS2 when glucose is present. We see almost 100% of ACSS2 moves into the nucleus when glucose is deprived. And then with this mutant that cannot be phosphorylated by AMPK, we see no ACSS2 in the nucleus in either condition. So we know based on these that AMPK phosphorylation of ACSS2 is required for the nuclear translocation. We see some of the images here. So wild type ACSS2, you can see it's almost completely in the cytoplasm. It doesn't, there's almost no co-localization with DAPI, which is the nuclear stain. And when the glucose is removed, so this is positive glucose, negative glucose, so when glucose is removed, you see almost all of the ACSS2 is now in the nucleus right here, and it's co-localizing with DAPI. Then we see the opposite pattern, where we see a defective nuclear transport. So with these S659A mutant that can't be phosphorylated by AMPK, we see no translocation to the nucleus, even when glucose is deprived. So this suggests that AMPK phosphorylation of ACSS2 completely prevented, or the mutation of this 659 residue completely blocked ACSS2 from entering the nucleus because AMPK could no longer be phosphorylated by AMPK. How then does AMPK phosphorylation induce the transport of ACSS2 into the nucleus? So to answer this question, they began with co-immunoprecipitations using these SFB conjugated Importin isoforms. So this Importin family is known to induce nuclear transport by the recognition of the nuclear localization sequences and then passes them off to Importin beta at the nuclear pore and induces the nuclear transport. So in this middle graphic, we see the results of a in vitro immunoprecipitation assay towards streptavidin. So they're pulling down streptavidin, which is conjugated to, in each of these columns, it's conjugated to a different isoform of Importin. And only Importin alpha 5, only this one right here, was binding ACSS2. So only Importin alpha 5 was successfully pulling down ACSS2 as indicated by the staining right here. So in order to see if this Importin A5 interaction with ACSS2 was facilitating the nuclear import, they assessed the nuclear localization of ACSS2 following Importin alpha 5 gene knockdown, indicated by this Western blot right here. So the KPNA1 shRNA, this is targeting the gene for Importin alpha 5. And you can see that ACSS2 staining in the nucleus was completely abolished by shRNA-mediated knockdown of Importin A5, because this is normal conditions right here. So when there's no shRNA and there's no glucose, we see the localization of ACSS2 into the nucleus. However, when you add the shRNA right here, you knock down Importin A5 and you see no translocation of ACSS2 into the nucleus. So the only difference between this last lane and this lane right here is the presence of shRNA, and it completely blocks ACSS2 translocation. So the researchers then identified a NLS, or a nuclear localization sequence, and they found it as this R664665A location, and they mutated it. So a lot is going on in this Western blot right here. Let's try and break it down. So we have 3 different histidine tagged forms of ACSS2. And this at the top, we have the R664665A, which is the NLS, and it's been mutated. So the nuclear localization sequence is gone in this top one. Probably should prevent it from binding Importin A5, right? And we also have the ACSS2 6 or S659A, which is the phosphorylation site of AMPK, right? So this should be preventing phosphorylation by AMPK. Then we have the simple wild type ACSS2. So we have wild type ACSS2. We then have a GST conjugated Importin A5 protein. So whenever you see a GST pull down, we're pulling down this right here, GST Importin A5. So we're pulling down Importin A5, and we're staining for ACSS2. So this entire Western blot is aimed at answering, also we have AMPK and AMP, right? AMPK and AMP. So this entire Western blot is designed to answer what factors are needed for ACSS2 binding to Importin A5. And this is ultimately indicated by the GST pull down, which pulls down Importin A5 because it's conjugated to GST, and then staining towards ACSS2. So they're doing a Western blot towards ACSS2. So let's look at the first column. We see wild type ACSS2 plus Importin A5, no AMPK, so we don't have that phosphorylation. We have AMP, but we see that there's no pull down of ACSS2, and this indicates that AMP is critical for the pull down of ACSS2, because in the second lane we see that wild type ACSS2, Importin A5, AMPK, and AMP. We see these are the basic components for the interaction between ACSS2 and Importin A5, because we see staining right here. It was successfully pulled down. So in the third lane, we're using an ACSS2 that lacks its NLS, so it doesn't have its nuclear localization sequence. It has AMPK, or no, it also lacks AMPK, but no pull down. Even with AMPK, still no pull down. So this suggests that the NLS is needed for interaction with Importin A5. And in these 2 columns right here, we see the phosphorylation site has been removed, and it doesn't matter what else you include. Even if you have everything else, all 3 factors, there's still no pull down, because even if you add AMPK right here, it doesn't matter because the phosphorylation site is missing on ACSS2. And then in the last lane, this is interesting because this is a wild type Importin A5 and AMPK. If AMPK is missing its AMP, it can't phosphorylate things. So this last lane right here just indicates that AMP is a required cofactor for AMP to function. So that was a lot. Let's go back over what we learned. So we learned that in vitro, all of this is in vitro except for the shRNA, that the interaction between ACSS2 and Importin A5 is critically dependent on, in the NLS, the phosphorylation to unmask the NLS is dependent on AMPK, the kinase, and AMP for the activation of AMPK. Okay, so at this point, you may have been thinking, okay, well, this is all in vitro. What does it actually mean in vivo? Does it actually correlate to in vivo processes? And in this slide, we see unequivocally that the loss of both S659A, the phosphorylation site, and the loss of the NLS, both of these mutations, these single mutations, completely block the translocation of ACSS2 into the nucleus. So we see in the wild type condition, glucose present right here. When glucose is removed right here, we see tons of ACSS2 in the nucleus. It's chock full in the nucleus. However, in these 2 mutants that only lack single residues, this lacks the phosphorylation site This lacks the NLS. Just doing that simply, it's not a big difference, but apparently it completely blocks its translocation into the nucleus. So it's a pretty important difference. You see, there's no translocation into the nucleus when these two residues are mutated. So in the next slide, we're going to be trying to identify what effect does the nuclear translocation of ACSS2 actually have on the physiology of the cell. So at this point, we know that ACSS2 is phosphorylated by AMPK, and it's bound by Importin A5 to induce nuclear localization. But what is ACSS2 actually doing once it gets into the nucleus? So to begin answering this question, the researchers immunoprecipitated flagged ACSS2, and this pulled down all of its interacting proteins. And then they ran all of these proteins on a gel, and they stained everything with Kumasi blue. And so in this first condition, there's no ACSS2. So anything getting pulled down is background static, getting pulled down by the flag antibody. This is what happens when there's no ACSS2. In this middle lane, we see ACSS2 with glucose. So this is the cytosolic interacting proteins with ACSS2. And this huge band right here, this is ACSS2 itself. And then in this last column, we see ACSS2 pull down when glucose is negative. So this fraction is what ACSS2 interacts with when glucose is gone during starvation. And we see a new pronounced band showing up at around 74 kilodaltons. So what is this protein at 74, 73 kilodaltons that's interacting with the nuclear ACSS2, but not the cytosolic ACSS2? It turns out to be the exact size of TFEB, the master regulator of autophagy. Indeed, the next step was to do an immunoprecipitation of TFEB and to see if it pulls down ACSS2. So that's what they do in these Western blots down here. So immunoprecipitation of TFEB pulls down ACSS2 during glucose starvation only in the nucleus, indicated by this band. So when glucose is negative during deprivation, we see a pull down and successful staining of ACSS2 right here. There's no staining of ACSS2 during glucose positive conditions. And we see in the cytosolic fraction, there's no interaction between TFEB and ACSS2, whether glucose is present or not. So TFEB only interacts with ACSS2 during starvation in the nucleus, indicated by this band. So their next step was to do a chromatin immunoprecipitation using an antibody towards ACSS2. So they're pulling down ACSS2, and then they're sequencing all of the DNA that comes down with it. So in the glucose positive condition, we see no DNA getting pulled down, which is not surprising because ACSS2 is not in the nucleus in this condition. And then in the glucose negative condition, we see the upregulation of a number of, the association of ACSS2 with a number of lysosomal genes. These are all lysosomal genes. We also see it in autophagic genes. They looked at that as well. Interestingly, and this is an interesting take on it, this R245-248A mutation, this is actually a TFEB mutation. So in these red columns, which you can't really see because there's no association of ACSS2, but when they mutate the NLS of TFEB, which is what this is, it actually prevents ACSS2 from binding DNA. So this indicates that when TFEB is prevented from getting into the nucleus by mutating its NLS, it also prevents ACSS2 from upregulating its genes. So we see that TFEB is required for ACSS2 association with lysosomal genes during glucose deprivation. And another thing worth noting is that 81% of the genes that ACSS2 targets and binds were also targets of TFEBs. So a lot of the genes, they share a lot of the target genes, and based on this co-immunoprecipitation, it's pretty clear that they're interacting with each other, and it's almost as if TFEB is taking ACSS2 along with it when it goes to these clear elements to prime gene expression. So the question now is, what is ACSS2 needed for at these autophagic genes? So we have this interaction between a known autophagy transcription factor, TFEB, and ACSS2. So what is the significance of this interaction? Well, considering that ACSS2 produces acetyl-CoA, a good place to start might be by looking at histone acetylation and acetyl-CoA concentrations in the nucleus. And so they observed that the nuclear acetyl-CoA fraction, the acetyl-CoA concentrations during starvation, are highly dependent on ACSS2 relocalization. So focus your sights on these red bars. These red bars indicate what's happening to the glucose concentration, the nuclear acetyl-CoA concentrations during glucose deprivation. And so they were looking at the amount of nuclear acetyl-CoA in four different conditions. So we see it in the presence of wild type ACSS2 right here. In the second column, we see when the phosphorylation site is mutated in ACSS2. And the third column, they see when the NLS is mutated in ACSS2. And this last one, T363K, this is when the enzymatic activity itself has been mutated. This is a ACSS2 enzyme that is no longer an enzyme and can't actually make acetyl-CoA from acetate. And you can see that under normal conditions, when glucose is deprived, so glucose deprivation causes the nuclear acetyl-CoA to drop, but not completely. So when wild type ACSS2 is present, you roughly have 4 picomolar of acetyl-CoA per this many cells. I don't think the actual amount counts or matters, but what matters is that the amount of nuclear acetyl-CoA drops off dramatically when either the phosphorylation site is mutated, or the NLS is mutated, or its enzymatic activity is mutated. So this collectively suggests that the nuclear acetyl-CoA concentration during glucose starvation is highly dependent on ACSS2's successful translocation into the nucleus. So next, they wondered if this has functional consequences for the levels of histone acetylation, because histones, remember, the histone acetylation requires acetyl-CoA. Acetyl-CoA is the cofactor that provides the acetyl group for the acetylation of a histone. So they performed another chip sequencing experiment, but this time they're pulling down acetylated histone H3. So acetylation's on H3. And they found that mutations in ACSS2 significantly reduced the amount of histone acetylation during starvation. And it was most pronounced at autophagic genes. In fact, when ACSS2 was deficient because of any of these mutations, there was basically no acetylation at autophagic genes in response to starvation. So this suggests that ACSS2's localization to the nucleus is critical for autophagic gene acetylation. And I should note that, although not shown in these figures, associated protein expression was also upregulated at these genes. So not only is there increased acetylation, but not surprisingly, there's increased mRNA and increased protein expression. So the acetylation of these histones by the upregulations of acetyl-CoA in the nucleus has very functional consequences for the expression of autophagic genes. Okay, so we just zoomed through all these fascinating interactions, but what is the functional consequence? Does it actually mean anything for a living organism? So this is the most amazing paper, this part of this paper, and I'm genuinely excited by these findings because I'm a nerd. So first of all, autophagosomes and lysosomal biogenesis in response to glucose starvation appears to require ACSS2 translocation. So, I mean, that's amazing. It requires, according to this paper, it requires translocation because you can see in these two graphs. Let's start with this one. So the autophagosome number per cell in wild type, we see the autophagosomes spike during glucose deprivation. That's not really surprising. You get more autophagosomes during starvation. But when you mutate the NLS or ACSS2's enzymatic activity, I'm surprised they didn't look at the AMPK deficient cell line that lacks the phosphorylation site. Either way, when the NLS is mutated or the ACSS2's enzymatic activity is mutated, you don't hardly see any increase in the autophagosomes per cell during glucose starvation. So we see only a very modest increase in the number of autophagosomes, and it is significant, but it's vastly less than when ACSS2 is wild type. We also see a similar deficiency in the lysosomal biogenesis. So we see in wild type ACSS2, the lysosomes spike following deprivation of glucose, as expected. But when the NLS is missing or the enzymatic activity of ACSS2 is missing, we don't see any significant increase in the number of lysosomes per cell. And so this is really quite amazing. I mean, this single mutation, just mutating this little NLS or preventing its enzymatic activity of this single enzyme, pretty much completely blocks the biogenesis of lysosomes and autophagosomes, presumably because the histones are not being acetylated. Remember in this slide we saw histones acetylated leads to upregulated gene expression, and without that upregulated gene expression, you don't see upregulated autophagosomes or upregulated lysosomes. So the next thing that they were going to look at is actually looking at the physiological consequences of glioblastoma cells to form tumors and to actually form tumors in the brain. So in these immunodeficient mice, so they used immunodeficient mice so they could transplant these glioblastoma cells and to grow them in the brain. And they noticed that the growth of these glioblastoma cells was significantly compromised by these minuscule mutations in ACSS2. So you see, this is the wild type. Wild type tumors or tumors with wild type ACSS2 grow huge, and they're slightly smaller when they're challenged with the 2DG, which causes glucose starvation. So this down here, this bottom row is glucose starved cells, and they grow pretty much just fine. But when these glucose starved cells have a mutant ACSS2, so in this lane, this small little growth down here, this is because the ACSS2 lacks its phosphorylation site. So we see the only ACSS2 enzyme present in these cells is one that cannot be phosphorylated by AMPK. So you see the amount of tumor growth is vastly downregulated in all of these ACSS2 deficient enzymes. So when you break ACSS2 by either preventing phosphorylation, masking its NLS, or destroying its NLS, or destroying its enzymatic activity, you see vastly downregulated tumor growth, and it's especially noticeable when the cells are challenged with the glucose analog 2DG. So It's really quite amazing. So these results collectively indicate through everything that following starvation, AMPK phosphorylates ACSS2 at 659, and this causes it to unmask at NLS that then binds to Importin A5. It translocates to the nucleus. It binds to TFEB, and then it upregulates acetyl-CoA at autophagic gene promoters, and then it promotes autophagic gene expression by acetylation. And this activity is actually critical for the survival and the growth of glioblastoma cells. I mean, for example, you look at this single, right here, these cells in this column, if you look at the difference between wild type and in this column right here, the only difference is that the ACSS2 enzymes present are lacking a phosphorylation site. So this single residue, this single mutation, vastly reduces the size of the tumors, and that's pretty amazing. So this paper in the end was really quite impressive to me personally, but one thing that does make me nervous is, are these findings applicable to other cells? So the current paper conducted all of their experiments in glioblastoma cells, which, although will have huge implications for cancer research, may or may not be useful to the rest of us. So cancer cells often behave much differently than normal cells, and confirming that ACSS2 is functionally important for autophagy in normal cells would be a major avenue of research, I think, to go from here. So if this activity of ACSS2 was shown to be a universal pathway, I think it would have huge implications for neurodegenerative diseases where autophagy is often not functioning properly. The study was also confining the autophagic stimuli to glucose starvation, but other upstream inducers of autophagy, such as amino acid starvation or growth factor deprivation, these are other inducers of autophagy, and I would be curious to see if they also signal through ACSS2 and trigger the nuclear translocation of ACSS2. But these are my only two major questions, and overall, I thought it was a very very nicely put together paper and very intriguing to read. And so I want to thank the researchers again for their contribution to science, and I hope everyone enjoyed going through this paper with me. If you have any lingering questions, please comment them, and I will promptly respond. Thank you.